somebody else, I will point it out. So as I said, it's a bit of a trip down memory lane. So actually this is me 20 years ago. Slightly shorter hair, that's probably the only difference. And then these are my roommates in Ferry Path and obviously the college. And so one thing I learned while I was in the Cavendish is that the works of the Lord are great. This is above the door at the physics department in the Cavendish down the road. Sought out of all of them that have pleasure therein. So seeking for pleasure in science is what my time in Cambridge was all about. And the person who actually got me to do that is Sir Sam, so I just wanted to give him a little ode before I start on the science, saying that this is actually what he was dressed like, come rain, come shine, on any day of the week. So he was a true gentleman scientist. And one of the things that was great about Sir Sam is that he liked lots of different things. And we were at dinner at the high table last night in Gomberlin Keys, and nothing, strictly speaking, nothing has changed. I think Rafi could agree with me. Except they don't have Stilton cheese anymore. It's the smelliest cheese there is, and apparently there's an art to figuring out whether it's quite gone off enough that you should eat it. Same thing for game birds, but these are all the things that Sir Sam liked, and he was able to invoke the love of gardening and the love of green peas, and the fact that different green peas, whether they're English or French, apparently makes a difference to their friction coefficient, which in turn makes a difference in terms of how they pack. And so 20 years ago, my whole PhD was related to saying, if we make mayonnaise, which is an example of frictionless packing, and if we make them transparent, then can we take a look inside? And that's basically what I did 20 years ago. And then Sir Sam would say, be that as it may. Some of you will recognize that sentence. That comes up a few times a day for him, or at least he used to. The last 20 years, I've basically been working on droplets, starting from this transparent mayonnaise and looking at contact forces and things we've been talking over the last couple of days, but then sort of expanding into biomimicry and self-assembly. And the reason why I'm spending any time on this now is because I wanted to make a nice parallel between my PhD 20 years ago and the PhD of my student Wen Jun Chen, who essentially managed to do better than what I did in everything she's done so far. So she came up with a droplet synthesis procedure that's able to make emulsions that are not only transparent, but simultaneously density matched. And that's something that I spent 1.5 years of my life trying to do and failed. I was just about to quit my PhD here in the Cavendish, and then we said, well, let's include gravity and make a different set of problems. So here's that actually working. So she managed to make buoyant and refractive index matched emulsions, and so this is based on a synthesis procedure which essentially grows a polymer. You choose your polymer of an oil, you essentially nucleate and grow it over time. You use some catalyst, you make sure that everything evaporates, you do it carefully, so we've done some perfecting of the actual technique. You stabilize with some surfactant, and what you find is that you can make droplets that are extremely well controlled, both in terms of the size, going from nanometer size droplets, hundreds of nanometers, all the way up to we can make 5-micron droplets that are perfectly monodispersed, and you can make them in large quantities. So it's pretty cool system for industry that you can actually control the size. So this is the difficult stuff. If you make a refractive index against a density plot, this is a ternary diagram of mixtures of oils, and the red line and the blue line are the aqueous phase mixes that you can make, either with glycerol or with DMSO. And so what you're really looking for is there a mixture where they have the same refractive index and density? And then the second layer of question is, can you get that precision to the 4th decimal place so that a 5 micron particle on the time scale of a week or two doesn't sediment or creep? So it's a very high precision, labor intensive measurement, and all credit goes to Legend. So here is the sweet spot that we chose to work. It's a mixture of these two oils. It's a mixture of the water with glycerol. And what she managed to do is pretty cool. This is a high volume fraction emulsion. You can completely see through it. If you put 5% volume fraction, it's completely opaque. You don't match it. And you can see how it's actually perfectly density matched over the time scale of a week to almost the 4th decimal place. And so here what you see is that a 10% volume fraction emulsion, so nobody's touching anybody, is purely suspended over the time scale of a week. And if you mismatch it by less than 1%, they sink or float, depending which way you mismatch. So you've got to be really precise, and she's done that now, so if there are industry people here, maybe you like it. The second reason why it's a cool system is because it's friendly to biological materials. So now you can take that same emulsion, you can functionalize it, say, with DNA. You can then make these guys aggregate, and what you find is that the adhesions between the particles or the forces in a later stage, you can actually visualize them in the microscope. So now you have it all. You have the adhesions, you have the structure, and you have no need to go into the space station anymore. So Paul Jakin should not get the mess of that, right? Maybe we should. Like, how can we stay on Earth and do the same experiments? Okay, so what can we do with transparent, buoyant, fluorescent emulsions? How can we put them to work? So this is what I actually came to talk to you about, and that is self-assembly. So if you think about self-assembly as a jigsaw puzzle problem, you want every single piece to maybe be a different color, a different shape, and a different interaction. And if you have that much information, you can build almost anything, given distinct species. But if you look at nature, that's not what nature does. We're not made of completely distinct species. We try to have minimal ingredients in which, for example, amphiphilic molecules self-assemble into membranes, in which amino acid chains fold into proteins, in which proteins interact, and RNA with proteins to make viruses, etc. So we want to learn what are the minimal ingredients with droplets that we can do to mimic life-like organizations. So that's somehow our motivation. And there's really two ways about it, right? Either you spend your time designing, for example, DNA origami and doing the jigsaw puzzle pieces, and you look for thermodynamically stable states which you've pre-designed about where you want to go. So you need a large alphabet, you need complex building blocks, you need directionality, you need valence control, deciding how many particles each one can touch, and shape. But you still get into trouble from a gas because you make lots and lots of mistakes if not every piece is distinct. So the way nature gets rid of that problem is to put everything in a line. And you'll find lots and lots of examples of that, starting from Alexandra's talk and beyond. And what's interesting about this system is that with just an alphabet of 20 amino acids, more or less all of life can be reproduced. So we say, what about material science? Can we scale up from molecules up to droplets? And can we then apply them to make geometries that are very well defined? So again, I wanted to show you this painting of Sassam in the ball, but he was famous for his beads on strings interpretations of what polymers do. And so staying in the spirit of things, now we combine droplets with polymers to make self-assembly. So again, this puzzle approach requires lots of information, but if you look at the matrix of interactions to make, for example, this particular shape, you need every single color to be different, and you need this many specific interactions to make sure that you get a unique geometric product. If you do the exact same thing and you limit yourself to 2 species, just blue and yellow, and if you then align them to begin with, what you end up with is that the blue-blue interaction is enough to make this one. So our whole question is, how much can we reduce the information and still get lots and lots of shapes out of this? Okay, so that's kind of what we've been worried about. We've called these colloidomers because they're chains of colloidal droplets. You can read about it, and the next piece of advice from Sir Sam was the first person into the bank vault steals the gold. There's even a book about it by Nigel Goldfeld, who collected thoughts about Sir Sam in this book. So the idea was, whatever we do in programmable folding from a 1D chain, it will be new. And so we had to design a game. What game do we want to play? So the game that we decided to play was to first make homopolymers, so droplets that are simply aligned with one flavor of DNA, and to then try to make heteropolymers that fold into specifically designed structures. So this is the work of Angus McMullen in the experimental side. The idea is to synthesize yellow and blue droplets. They're bound together through these specific DNA links. They have one interaction that sets the backbone. Even if you heat up the sample to almost boiling temperature, 75 degrees, they won't come apart. As you cool it down, there are other weaker DNAs that you sort of see here, and they come into play when you cool the system down. So all of a sudden you can have a hierarchy of interaction driving you through the landscape into some misfolded state in this case, and that's a whole other era of how to rescue and chaperone molecules out of dead ends. But that's another topic. Okay, so what's this based on? This is based on the idea that there's a very well-defined sharp melting temperature above which all the DNA comes apart and below which all the DNA sticks together. The idea is that you start at a high temperature and you switch on sequentially the different interactions, say blue likes blue, and then yellow likes blue. And the question then is, just with an alternating sequence, how much can you actually make? And so what we decided to do was to look up this huge library, which Michael Brenner's group started with Miranda Holmes-Safan and Zorana's previous work in her postdoc. They enumerated all the possible geometries that clusters of n particles can go into. So forget about the chain, just ask how many rigid configurations are there for, say, a chain of 7 droplets. What you find is that in that case there are 4 different geometries that you go into. And if you run the free energy landscape of this guy and you compute it, you can see that they have different free energies. But the trouble is that you always get a mixture of all of these. The kinetic traps are too important. So you really need more than just thermodynamic equilibrium to drive you. You need design to actually select even the more stable state, but you also then get access states that otherwise wouldn't be possible. Right? Design is needed. So this is what we do. It's a kind of pruning method. Here you see a potential energy landscape in which you start with a chain. Each time you add a bond, you go down this tree. Obviously, the more bonds you add, the more possibilities there are. As you rigidify, the less possibilities there are. So this is somehow a measure of entropy of the number of states you have for a given number of bonds. Then you cool it down, and what you find is that instead of the gray tree, which is everybody likes everybody, you end up with pruning it, and you only get a few trajectories that can lead you, say, into yellow likes yellow. And if you then switch on, in this case, blue likes yellow, you end up with this Christmas tree, and then you can lock it in with blue likes blue finally. Okay, so the whole point is you can do a search algorithm within which you can identify which protocols lead you to a unique structure, which we're going to call the fault. So that's the game we decided to play, and I'm not going to talk about it in too much detail, but just to tell you that instead of enumerating the entire tree and having very expensive computational trial and error mapping, what you can do is you can essentially go backwards. You can identify, you can start with the rigid structures because you know those, and then you can identify backwards where the local minima are. So let me explain that in a little bit more detail, because if you start with a colloidomer, you make this graph theory of the graph representation of the rigid structure, and then you have a Hamiltonian, which is the number of self-avoiding random walks that you can draw inside of this structure. So you actually know how many ways you can construct that particular fold. And after you enumerate those Hamiltonian paths, you can essentially add them the colors that you know. In this case, just blue, yellow, blue, yellow, blue, yellow. And then you can, in a backward fashion, add blue likes blue, and those would be the secondary structures. And you can simply ask, are you rigid or floppy? Are there any more bonds that you could form with yellow, blue? And so you prune this landscape in a backward fashion. So this is just a trick to reduce the computation time, nothing else. It's just a way to get to the bottom of the tree. Okay, in terms of the results, they're good and bad. The bad news is there's 1,000 rigid structures for chains up to, say, n equal 12 droplets. And then you can ask, how many of those thousands can you uniquely get with these protocols? And with just two colors, there's 12 solutions. So you're looking at about 1% yield of foldamers in this sea of possibilities. And that's what the algorithm gives you. And what's nice about this is that you notice that, for example, blue likes blue, yellow likes blue, yellow likes yellow has lots of solutions. So let's say you start with a soup of droplets that has all sorts of chains. You can see that this protocol, by the way, droplets with 3 or 4 or 5 chains only have one geometry in any case, so they don't count. So if you look from 6, 7, 8, 9, 10, 11, the 12mer doesn't have a solution, so that's a bummer. But up to 13, this one protocol, you start with a soup, you pull it down, should give you a unique solution for every single n other than n equals 12. So that's a nice protocol. Then other protocols give you a few more. Okay, that just says what I said. So how do we actually do this? This is for the experimentalists among you. And actually this is where we spend the bulk of the time. We need to design DNA that doesn't melt at 75 degrees. We then need to have 8 base palindrome interactions, so that's where the base pairs read the same forwards as they do backwards. That means that they're homophilic. So these guys melt at 40 degrees, and if you make shorter chains, then you cool it down. So what's a way that I can trigger the DNA to interact such that there is no overlap? That is, all the blue-blue bonds will form before I start with blue-yellow. That's very important to not make mistakes. So we spent a lot of time optimizing that, and this is actually raw data from the microscope, and what you will see is that, sorry, I have to actually make it run, what you'll see is that we end up with 100% yield in a lot of the time for the predicted pathway. So that was really nice. It meant that the experiment is working, it meant that the algorithm is working, and it meant that we could make these crowns, we could make these triangles. And what you'll notice is that not all of them do as well as the others. So for example, the crown only has a 38% yield. Doesn't mean that the rest of it are mistakes. The rest of the ones that we don't call success stories are the ones that get trapped in local minima. So instead of looking at downhill folding, you can do a much more elaborate scheme in which you can optimize the temperature such that it's not purely downhill, but there is a rate optimization so that the backward rate is just enough that it actually gets you into the track that you want. And so we've been playing with a totally different project on the theory end, which is how can I have a protocol in which I vary temperature in non-trivial ways such that the backward and forward directions are allowed along the tree. And so here is a pure downhill simulation, and here is where rearrangements are somewhat possible, and you will see how this one sorts itself out, or this one does. So this gets into the game of chaperones and how you might see the right geometry. Okay, so what did we learn? We learned that in our case, this is my favorite slide, experiments do better than theory in the sense that these little guys are not density matched, and so they're sitting on the surface, but they can have 3D excursions every now and again. And so that's already a rescue operation. So local minima that cannot resolve in the simulations in 2D actually get there in our experiments. And so these out-of-plane flips, we get 100% yield in the experiment, not so in the simulation. So what did we learn? We learned that all the 12 guys that actually fold do it via the same mechanism. They first form a hydrophobic core, and then they lock in the outside. And so that's kind of similar to the hydrophobic collapse of proteins, where you first get a crunch, and then you sort out the outside. There's a second, less likely solution, which is that you make a circle, you kinetically trap the system, and essentially the only secondary ones that can be made are within that. So we call that the kinetic, the geometric frustration mechanism. Okay, so how much longer do I have? Because I did want to show
5 minutes on the question.
Excellent. So I wanted to show you a couple of new things. So now you can extrapolate from this. You can say, okay, I've convinced myself I can do programmable design. I can add more letters. I can scramble the sequence and pretend that I'm an evolving protein and that I'm now playing around with genes. And what you find is that you can now have a much bigger playing field. Instead of 12 folders, we've got, on the order of a few hundred. And so with these new structures, you can actually start to ask questions like, what 3D geometry can these 2D folds fold into? You can start to ask questions like, can I make a hierarchical assembly to make a quasi-crystal? Can I start to mix folders of different types? And okay, so up to n equal 9, we can encode everybody in 3 colors. That's pretty cool. Above that, like I say, we have, I think, 385 winners or something like that. And then you already start seeing that really cool things can happen. If blue likes blue for this guy, you're going to end up building tubules. This is probably, how many of you are old enough to know about Space Invaders? These are Space Invaders. They only make dimer complexes, so they can't do anything else except find one partner and then they're done. So we start to look at complexation as another route. We managed to make it work in the experiment a little bit, but the bar is much higher in the experiment now than it is in the simulations. And here's the trouble. This is our worrisome slide. When you're in 2D, things look kind of doable. This is the chain length. This is the number of rigid configurations. But when you start to have an HP model, say where you have two colors, the number of folds is just start to explode. And so you can ask the question, how many solutions are there in going from 1D to 3D with two letters? And I think the answer is like 2. We found 12 in 2D, there's 2 in 3D, and they're only for n equals 6 and 7. So let me show you, because this is where I'm combining the density matching and the refractive index matching with the programmable design, so this is pretty cool. I don't know if you have a good enough eyesight for that, or if the images are good, but you should be able to see that there's a little tetrahedron forming here, and it's got a bunch of stuff. It basically locks into a stellated tetrahedron, so we do manage to do 1D to 3D folding. For the 7, this one is also cool. It's a stellated tetrahedron, we can make it. But now the question is, what's it good for? Now that you can make these little 3D geometries, we wanted to make mayonnaise robots. So we said, how can we actually open and close these architectures? So we managed to make them, but can we actually do some function? Because now we start to build up towards the dogma of biology, where you start with a sequence, you have some design of a geometry, then you have some function, and then you can feed it back and say, can I optimize the sequence to get a better function? So we wanted to have that whole thread, and this is the coolest slide. Oh, publish and be damned, that's the last piece of advice from Sept. You can, okay, I'm just going to summarize. If you had a way to do logic gates in which the DNA that binds the droplets could be by CRISPR or other enzymes or the pen toolbox, some of you might know, if you could produce it in space and time using these enzymes, then you can have spatiotemporal control of what these little geometries will do. And so Lucas Denier, who is the new postdoc in the group, has done some really beautiful work. So I'm just going to show you the final result, which is that he starts with a gas of blue and yellow droplets. He then has these enzymes that in time produce the sticky DNA that bind them together. He then later on has a nickase, which is a DNA that comes in, an enzyme that comes in and cuts the DNA. And so what you should see here is that the clusters not only form, but they also, not only form, but they also dissolve. They can form and they can dissolve. And so our idea is to try to make these rigid geometries dynamical through these logic gates. So I'm going to end there and just thank my group again. Thank you for listening, and thank NSF for funding.